All right. Good morning and afternoon and very, very late night to some of you. Thank you so much for joining in today. We will be presenting today big team science projects in the majority world, and these are projects led by scientists from the majority world. Um, a lot of the times we ask ourselves, why is this important? Uh, it is important because most big team science projects and thus publications are dominated mostly by early career scientists in the minority world. This is a publication by Buchanan and Lewis, showing that most projects are originating from the US. And as we all know, this can influence what type of questions are being answered, what types of methods are being used, and the samples that are representing this. But we need more projects that are being led by researchers by the majority world. And here we will be presenting three projects of that sort. One of them is by a researcher from Serbia, and that will be talking about drivers of questionable health behaviors. And the other one is from a researcher from the Latin American Genomics Consortium, where we are trying to improve representation of psychiatric genetics research. And the last but not least is another one led by a graduate student from the University of Arizona that to full disclosure, used to be my undergraduate student in Mexico. So the three presentations, we will start with Amelia Ibarra Mevans, who is currently a graduate student in the US at the University of Arizona. And we will start in order of how far along the projects are. So this is a project that is just starting, and it is Vigilance in Latin America, methodological insights from international questionnaire translation and data collection. We will then move on to a presentation from Diana Núñez Rios, who's originally from Colombia but is now at Yale, and will be presenting a project on decoding rare CNVs and their functional insights in major depressive disorder from Latin American cohorts. This is a working progress, but she has some preliminary data already. And but last but not least, Iris Zezelj from Serbia, from the University of Belgrade, will be presenting In Search of Universalities in Health Behavior, what we learned from HEALTHQuest team, which is right now in the process of being sent out for publication. Sent in already, hopefully will be in the review process pretty soon. So with that brief introduction, I will let Amelia take it away so that she can start her presentation.
Perfect. Thanks, Nadia. I'm going to be sharing my screen. Okay, perfect. So hi, my name's Amelia. I was a previous student of Nadia's, but I think mentoring and supervision never ends. So I think I'm still your student. And we still get the pleasure to work together in projects like these. We call this the Vigilance study, Vigilance in Latin America. And I want to talk a little bit about my background and how I got where I am. I am from a little town in Mexico called Hermosillo. It's on the north west side of Mexico. And if you drive 4 hours north, you get to Tucson, which is where I am now. I did my undergrad in my hometown. I had lived there my whole life, and then decided I wanted to do grad school. So with a mix of skill, opportunity, luck, and lots of help from Nadia as my undergrad mentor, I went to the University of Arizona, which is where I am at now. I am in my 4th year of the PhD program, and I'm doing clinical psychology. So it's been an exciting journey. So here's a little bit about of the agenda for today. I'm going to be talking about the scientific background, why vigilant study, a little bit of the methods, and I'll talk about the current status of the project and what are some challenges and strengths from this project that we ran into. So let's start a little bit with the scientific background. So when I moved to the US, one of my main goals was to continue studying Hispanic Latinx populations because they were, or we still are, so underrepresented in literature. And one of the interesting phenomena that I learned about when I first got to grad school was the Hispanic health paradox. So despite being exposed to many socioeconomical stressors and environmental stressors, we Hispanics have a mortality advantage. So we are living longer than non-Hispanic whites, but also have slower progression of disease, for example, in cardiovascular disease. And there are many theories as to why that might be happening. One of them is the sociocultural and the language hypotheses, which talks about how Hispanics have greater social cohesion, bigger social networks, and also language has this buffering effect on stress and other related processes that may be translating into that mortality advantage. So I found that fascinating. And one of the ways that I conceptually think about how that may be playing out is through social vigilance. So social vigilance is the active behavior of monitoring the environment and look for threats. So it can also be understood as the appraisal process of Lazarus and Folkman's stress theory. So we're constantly appraising our environment and look for threat. And one would think that Hispanics, Latinos, being in a disadvantaged position, they might have higher social vigilance. However, I found in my master's thesis that that was not the case. Actually, Hispanic Latinx participants were reporting lower social vigilance. So there's this disconnect between how much they're exposed to in terms of stressors and how much they're actually experiencing. So that became kind of like the driver for this study for me. I wanted to see how those results would generalize because this is not only important for Hispanic Latinx participants living in the US, but also the majority of Hispanics are located across Latin America. So I wanted to see how those results would generalize. And if there were differences, where those differences lied. So to do that, we partnered together with different universities. And first of all, we did translate the social vigilance questionnaire, which is kind of like the gold standard measure to measure vigilance. Through that process, we again partnered with different universities. I myself at the University of Arizona, University of Sonora in Mexico, a university in Peru, Colombia, and Chile. And we all got together and talked about the measure and tried to get at a version that was usable and culturally appropriate for the different sites. Because even though we all speak Spanish, there are certain nuances in the language that vary by region. So we got together and translated the SVQ with also an incredible team of research assistants that helped us through the whole process. And once we translated the measure, we also included measures of mood and anxiety, symptom stress. And because we're all interested in health, we included measures of sleep, long COVID-19, healthcare mistreatment, but also personality and affect. Participants who are eligible to be in the study are those who are at least 18 years old, and they have to be fluent in either English or Spanish, given the questionnaire is available in both languages. And the data will be collected using REDCap And overall, the survey will take roughly 45 minutes to 60 minutes to complete. So it is a very comprehensive survey. It is quite long, and it tries to capture many of these behavioral aspects of health, but more importantly, these processes of stress exposure, vigilance, and how those may affect health in Hispanics. The current status of the project is we have roughly about 1,000 participants. Chile and Peru are yet to start data collection because one of the things of doing an international study is, again, reviewing ethics and IRB. So they're going through that process right now. But we have about 1,000 participants, 1,028 in the US, 75 in Mexico, and 11 in Colombia. So there were many challenges as well as strengths of this type of study. One of the challenges was definitely translating the social vigilance questionnaire. We had to ensure cross-cultural validity, and again, ensuring that the Spanish version was acceptable and reliable for all sites. And then one of the things that I think many of you might run into is the recruitment systems. So in the US, in certain universities, there's a pool of participants you can access through the university of those who are in Psych 101 classes. They need research credit, so they participate in different experiments or research studies. So we got our participants in the US through that system, and it's a very straightforward, you get a lot of participants that way. But of course, those systems are not available at the other sites. So that's why the sample looks much bigger in the US. In Latin American sites, it's more like word of mouth kind of thing or recruitment via email, which tends to be a little bit slower. But nonetheless, we're again trying to ensure quality and validity of the study. So it's okay that it's just coming in slower. We are taking into account that those recruitment systems are different. Another challenge that we ran into was funding. We initially wanted to compensate participants for their time. But again, currency and international status was just difficult to get the money to our collaborators so that they could compensate participants. We thought about using Amazon gift cards, but then one of our incredible research teammates was like, well, if I could actually have the money for my participants, they might use it for groceries, which is a very real thing that we had to consider. So funding has been challenging. And of course, survey development, trying to find all of the measures that we usually use for our studies in the US, trying to find the measures in Spanish, but not a Latin Spanish, not a Spanish from Spain, or something that would not make sense for somebody who is in Chile, for example. That was quite challenging. However, I also like to talk about the strengths of these kind of studies. First of all, we had an incredible bilingual team. Everyone in the team spoke English and Spanish, so we were able to again look at the measures and kind of decide if it made sense, compare them, but also our US team felt very included, and it was just a great environment of collaboration. The generalizability of the results will be important to address once we finish the study. I think comparing social vigilance scores, but also overall scores in general throughout the survey and how these relationships play out is going to be really important. And then the advantage of the study is that we're not only focusing in Mexico, but also in different regions of Latin America. So those cross-cultural comparisons will be possible once we have the data. And we had a very, again, comprehensive survey. It was very lengthy, but also very capturing a lot of aspects of behavior. So that's going to be, I think, one of the strengths of the study once we finish data collection. So we are hoping that these findings will extend the Hispanic health paradox literature by not only focusing on how those relationships play out and those psychosocial mechanisms play out in the US, but how those generalize in Latin America. And I believe that's important to provide those cross-cultural insights into disparities in health and mental health in general. Yeah, I just want to say thank you to the whole team. You may see some familiar faces. I want to say thank you to our incredible research assistants and my lab, my mentor, and everyone else who contributed to this study. Thank you.
Thank you, Amelia. That was very insightful, and hopefully people can get a little bit of to use the same word, a little bit of an insight what it is to do something in Latin America. So we will leave some time at the end for questions. So hold on to your questions, and we will let Diana take the lead now.
So in these 20 minutes, I'm going to talk about copy number variant analysis in Latin American cohorts with a particular focus on depression. This project is, as Nadia said, is a work in progress that we have been able to do thanks to the Latin American Genomics Consortia that is led by my PI, Janitza Montalvo-Ortiz, Paola, and Elizabeth. And this consortia, we have monthly meetings when we try to discuss and improve the representation of Latin American samples in the genetics or in the genetic research of psychiatric disorders. We have different working groups, and that's why we were able to collaborate and to have some samples to perform this analysis. So I will give you a brief introduction about depression. Depression is a complex multifactorial disorder, which means that there are many factors that can increase the risk of developing this disorder. Some of them could be environmental, like for instance, exposure to stress or trauma. Some of them are biological processes that have been already related with this disorder. How our body could respond, for instance, to trauma exposure, and can increase the risk of develop psychiatric disorders. And I'm going to be focused in this talk in the genetic part, because there is also some genetic information that can increase the predisposition to develop this psychiatric condition. So I don't know how many of you are familiar with GWAS analysis, white association studies, when the idea is to evaluate the genome, how the genome could increase the risk of developing psychiatric disorders. So in general, we have the genetic material that we inherit from our parents, and we go to the smallest part of the genetic material that is called nucleotide. And this nucleotide could be usually referred like with 4 letters, GCAT. And the idea is to see the representation of these letters in cases and controls, and then after a statistical analysis, try to find those letters that are overrepresented in cases compared with controls, and try to find those that are associated with the condition that we are evaluating. So this nucleotide could have maybe a functional impact, because we need to have a perfect combination of this letter to be able to produce an amino acid, and a perfect combination of amino acids to be able to have a functional protein. So if there is a disruption here, we could have some consequences. Some of them we could not have consequences, but the idea is to try to identify those nucleotides that will increase the risk of this disorder. So if one nucleotide could have that impact, we hypothesize that structural variants that could involve at least 50 nucleotides on the same genomic region maybe could have a larger impact. So one of those structural variants corresponds to copy number variants. So hypothetically, we should have 2 copies of each gene, right? One inherited from our father, one inherited from our mother. But it's completely normal that across the genome, we will lose genetic information and we will get extra copies of genetic information. So for instance, here, we can lose the 1 copy of the gene C, and we could have 3 copies, 1, 2, 3 of the gene D. So we will need to identify if those changes could be just normal or could also increase the risk of developing psychiatric disorders. So what do we know about copy number variants in depression? There are already a list of copy number variants that have been highly recognized in neurodevelopmental disorders. And there is already known that those copy number variants not only increase the risk of developing neurodevelopmental disorders, but also schizophrenia, this is part of that, but also depression. So the list that you can see over here is kind of the address of those copy number variants. The first number is the chromosome, and the other numbers are the location in the chromosome. But as Nadia was talking at the beginning, most of these genetic studies are focused particularly in European ancestry. What does it mean? Where is our genomic coming from, or genome coming from? So most of the individuals that have been participating in these studies across different disorders are from European ancestry. And this is a figure that the LAGC paper published this year, when the idea was to evaluate how is the status of Latin American samples in this research. Most of them are Latin American living in the US. There are some Latin American studies that are local, performed in Mexico, Colombia, Brazil, Peru. And when we go through the disorders, there are some studies that have been focused on depression, PTSD, or substance use disorder. But the representation of Latin American samples is still low. So trying to increase the representation, that is one of the goals of the LAGC, we were able to collaborate with researchers from Mexico, Colombia, Puerto Rico, Brazil, and also Latin American population here, researchers here in the States. The results that I'm going to show today are focused on these 3 cohorts from Mexico, Colombia, and Puerto Rico. We have a slightly higher number of females compared with males, and we have a representation of cases compared with controls. But the idea is to include more cohorts and also try to have more similar sample sizes. So why is important to evaluate Latin American samples? Because I was telling you that we could be quite different from the European ancestry. This is an example of that. We have in blue African ancestry, in yellow European ancestry, in red Native American ancestry. And when we see how is the distribution of those ancestry in Latin American countries, we can see a mix of that. There are different proportions of each of these ancestries. What does it mean in our genome? That exactly in the same chromosome, we could have a particular region that is coming from African ancestry, another region that is still part of the Native American ancestry, and another region that is part of the European ancestry. This mix was after the colonization process, when European and African ancestry start mixing in these countries. So when we evaluate the ancestry, there is a big project, 1,000 genome projects, when they collect samples from different parts of the world, and we can use those samples as a reference to try to understand what is the ancestry of the samples that we are working with. So here in the bottom are the colors of the Latin American cohorts. In yellow and blue are the Mexican and the Colombian cohort, that you can see that the distribution is highly close to the Amerindian ancestry. And the Puerto Rican cohort, that is also Latin American, you can see that the difference, the distribution of this ancestry is slightly different. So this is to highlight that we are not only different from the European ancestry, but we also have a lot of differences even within Latin American countries. So understanding the value of these samples, we are collaborating with the PGC, Psychiatric Genomic Consortium, that they have monthly meetings also to discuss about how to improve the analysis of genomics in psychiatric disorders. And we are collaborating particularly with the copy number variations working group leadership that they already have the pipeline established to perform an accurate CNV calling. So as part of that pipeline, this is the software that we use to do this CNV calling. That the idea is that this is like kind of the reference software. Sorry. When we need to get information about the letters that I told you at the beginning, and also the intensity of those letters. So on that way, we can identify if the genomic region have 2 doses, or we have 3, or we have 1. But to improve the accuracy in this analysis, we need to perform the CNV calling by the second software. That could be QuantiSNIF of GenoCN or others. And the idea is to work in the downstream analysis, work with those copy number variants that were called by both software. We also perform a QC where there is already, let's see the distribution of that data, that we don't have outliers that can make noise in our analysis, and also to remove some copy number variants that we already know that are part of the center of the end part of the chromosomes that could increase the noise in the analysis. I won't have enough time to show the distribution of the copy number variants that we found, but in general, we found more deletions than duplications, and no representation of copy number variants and larger sizes in cases compared with controls. But we need to remember that we are working with a larger samples of cases, so we need to make sure that this data is because of the disorder and not because of the sample size. But for now, I can show you how is the CNV distribution of Latin American samples compared with the ones reported in public databases. This DGB and NOM-AD, they have information of copy number variants that have been reported for different groups, most of them based on European ancestry. So as I was explaining before, we are an mixed population. So the idea was to compare which of them had been already reported in these databases. And we can see over here that most of the copy number variants that were found in the Latin American cohorts had been reported in these public databases, DGB, NOM-AD, or even in both. But there is about 10% of the samples that were reported that were only found in the Latin American cohorts. So it is showing that we have a population stratification of those copy number variants as is expected. To try to understand better what could be the biological role of these copy number variants that we are finding in the Latin American cohort, there is a measure that is called PLI. This is a score that ranges from 0 to 1. When it's close to 0, it means that it's tolerant. It means that we can get extra copies, or we can lose that genetic information and nothing will happen. There is the ones that are around 0.5 are those that are hard to interpret because some individuals could be tolerant and other ones no. And those that has a score higher than 0.9, it means that if we get those changes, most likely we will have a functional consequence. So we wanted to see the distribution in terms of that tolerance. We have only copy number variants that were only found in Latin American cohorts that are tolerant to those changes, but also some of them that are not tolerant. So to have a better idea of this biological impact that those copy number variants could have, we can see that those copy number variants that are mapping genes involved in neuronal process or brain development correspond to deletions, loss of genetic information that are not tolerant to those changes and that were found across different ancestries. But when we focus with the same type of copy number variants that were only found in Latin American cohorts, we can see that also there are some of those copy number variants that could be involved in brain development and also in a lot of regulatory biological process. And here that this is enrichment analysis, those deletions, no tolerant that were found cross ancestry were enriched in unipolar depression, and those that were only found in Latin American cohorts had been related with developmental disorders, which some of the copy number variants associated with neurodevelopmental disorders had been also associated with depression. There is another strategy to evaluate the role of copy number variance in these complex disorders. It is something that is called copy number variance risk. Like the idea is, I told you that we have a PLI score. We could have an idea of which could be the functional impact of this copy number variance. So the idea is to see like the total score across all the copy number variants that each individual could have. This is work in progress, but when we we evaluate each cohort separately because we are having differences in ancestry levels, and then we meta-analyze the results. Meta-analyzing those results, we observe that there are some sex differences in the CNV risk score in depression, which could make sense because the rate of females affected for depression compared with males usually is higher. So we kind of expect at clinical level a higher rates of depression in females compared with males. And it is something that we are also observing here in the CNV risk score. But when we perform the stratify analysis, evaluating only females and only males, we lost that significance. But what we are observing here, that this is preliminary data again, but the Z-value for females is positive, for males is negative, which means that those CNV risk score could have an opposite impact in both sexes. We also perform this CNV risk score only focus on deletions, only focus on duplications, and we found a significant, in the stratify analysis back sex, we found significance with the duplications. So we will need to start filtering those copy number variants and try to understand better what could be the role of those CNVs in depression. But that will be later. So as a conclusions of this part of the analysis is that we confront the complex Latin American admixture, the ancestry differences even within Latin American populations, the CNV population stratification, that although most of the copy number variants that we found in the Latin American cohorts have been reported in public databases, both European-based databases, there are about 10% of the CNVs were only found in Latin America, and it looks like that some of them have important role in brain development and different neurodevelopmental process. And based on the CNV, the preliminary CNV risk score results that we have, we could think maybe that the copy number variants maybe also play a key role in the MDD sex differences. The next steps will be including additional LAGC cohorts. We'll be performing the CNV GWAS. So that way, when I show you the Manhattan plot with the SNPs, we want to identify which particular copy number variant, the particular address of this copy number variant that could be increasing the risk, contributing to depression. I want to clarify that each of those SNPs or each of these copy number variants no carry that particular genetic variant will be enough to develop depression. It's just something that is adding risk to develop this disorder. And also understand better about population stratification, understand better what is the role of those genetic variants that are found across ancestries and those that are found only in the Latin American cohorts. And the acknowledge for the Montalvo-Ortiz lab, my PI that, as I said, she's also leading the LAGC, members of the LAGC MDD working group, and the researchers that collaborate with their data, the PGC CNV that helped us to develop this analysis. That's it. Thank you.
Thanks. Thank you so much, Diana. This brings into light a lot other barriers and things that might make it more difficult. Last but not least, we'll have Iris talk to us about very exciting project that is in its process of being in the process of publication, hopefully pretty soon. So take it away, Iris. Thank you.
Thank you so much. So my name is Iris Zezelj. I teach the social psychology at the Department of Psychology, University of Belgrade. I'm very happy to be here and thank you for inviting me. What I'm going to share today is the lessons learned from this large big science team called HEALTHQuEST, mostly out of psychologists, but also people from different social sciences and some with medical background. And what we gathered together to do is search for universalities in health behavior, a very ambitious title, but I'm going to guide you through to see what we exactly mean. So let me see how I'm going. Just a second to see how I can go through the slides because this is not working. Okay. Okay, now it's working. Great. So here is the team. I'm not going to go through all the names. You see, there is a lot of us, and unfortunately, we haven't met yet, so we don't have a group photo like you guys did to share. But we come from 33, hopefully at one point we will, at least partially. So we come from 33 countries across 5 continents, and I'm very happy to say that we are a very, very diverse team. So why we gathered together to study health choices? First of all, we make tens or even hundreds of health-related choices every day. And while most are automatic and guided by habit or situational cues, there is a portion that are intentional and involve deliberation. And from psychological point of view, there are some that are particularly puzzling. And those are intentional health choices that put one's health at risk. So that's what we actually gathered together to look into. We labeled those, and those are labeled like that in the literature already, questionable health behaviors, and put them into 2 broad categories. One is intentional non-adherence to the medical recommendations, which is self-explanatory, not good. So it leads to high mortality, spread of infectious disease, poses burden on economic burden on health system, and so on. And public health crises, such as recent pandemic, make it painfully obvious that we need to establish cooperation with the public so they can intentionally reach the decision that's, so to speak, right. So they intentionally decide to adhere and not to adhere. And that this is not only crisis related. For example, vaccine hesitance and antibiotics overuse have been constantly listed in top 10 global health threat by WHO. So there is that. And on the other side, people can turn to non-evidence-based medicine. They can use traditional complementary alternative medicine, TCAM in short. And it's contrary to the widespread assumptions, it's not harmless at all. So it can have side effects on its own, but also it can lead to avoidance or postponing medical treatments, interact with those treatments. And there is, for example, evidence that cancer patients who first turn to TCAM instead of official treatments have significantly shorter lifespan. And there are even deaths and injuries documented for the mildest herbal products such as aloe vera or mulberry leaf and so on. And what we also see is that both categories of behaviors are on the rise, and the rise is sharp. We labeled both of these categories of behavior intentional to distinguish them from unhealthy choices that are automatic, a result of lack of resources or willpower, and to emphasize that they require actively engaging in non-recommending behavior or avoiding or modifying the recommended ones. And if we dig deeper, as we psychologists tend to do, to see what intentions shape this particular behavior, we see that it's not shaped by a strange wish not to be healthy or to be unhealthy. So there is not like this portion of population that behaves in this weird way, but is a result of a specific worldview that makes these questionable behaviors look like good choices, like best and healthy choices. So we wanted to see whether first that's true, and then if yes, which of these beliefs are mostly correlated strongest with the questionable health behavior. And what we did 5 years ago in Serbia is we applied for funding in local Ministry of Science, and we got fund for something called Reasons for Health, where we did several things. First, we scanned the terrain. So we tracked the questionable health practices and mapped them to a group of unfounded or irrational beliefs. And we also analyzed media landscape around these TCAM practices. Drawing from that scan, we created and tested different communication messages that aim to reduce the questionable health behaviors. And we also wanted to initiate social dialogue and involve the stakeholders, general public, chronic patients, healthcare workers, educational system, and media in the dialogue on these issues. So we only initiated that. I cannot say that we were super successful, but we started with that as well. And we published a bunch of papers with the results from these projects. We first developed the instruments. Those were retrospective checklists that marked both intentional non-adherence and non-evidence-based health practices. We started with a very large pool of behaviors, hundreds of behaviors, and narrowed them down to the most typical ones. And we made that checklist in which participants are asked to mark the behaviors that they engaged with in a certain time span before. So we published both, and we also looked into this psychological footprint of all these behaviors, and we found very robust and consistent results. But the fact is that they were tied to Serbia mostly. We did some replications in Bosnia and UK, but mostly what we had as a response from reviewers, and we had to discuss it back and forth, is whether this is like a peculiarity of Serbia in terms of the behaviors themselves and also about their psychological footprints. So we needed to argue that even if that's the case, that would also be relevant. But we also had this feeling that this might be culturally universal and that these results would hold cross-culturally. So we were kind of nudged or pushed to test that. And we put out a call for collaboration where we said, listen, we are a team of researchers from Serbia. We have these instruments to measure questionable health behaviors and their footprint. Would you be willing to join us in this collaboration? We offer co-authorship in return for collaboration. No resources there, like zero. We have nothing to offer you in terms of resources. And we would have been happy if we had like 5 or 10 laboratories coming back, but what happened exceeded our expectations by a large margin. So as I mentioned at the beginning, we had more than 33, but this is where data collection has been done and successfully done. 33 countries, 5 continents, and a database of more than 15,000 participants. So at point, it was really overwhelming to see like data just coming in. Now I'm going to guide you through quickly the results of the project. So first, let me just address this cross-cultural prevalence of these questionable health behaviors. What we see beyond a doubt that they're more of a norm than an exception. So let me illustrate that. So for example, lifetime engagement in at least one of the listed questionable health behaviors was close to 100% in all samples. That means that we couldn't find almost a single participant who did not engage in at least one of these listed behaviors. So it's not a very strange or like a fringe type of behavior, but it's basically a norm. And let me now focus on these 2 categories for this intentional non-adherence. For example, like I took an antibiotic even though a doctor did not prescribe it to me, or decided to stop taking antibiotics earlier than prescribed when my symptoms were relieved, for example, or decided by myself which of the drugs I should take and which I should not, and so on. So we averaged those behaviors, and here's what we see in these large samples. So the average percentage varies between 30 and 40. So it's really high, and it's not, we do not observe any striking cross-cultural differences. So it's quite common, and it's common worldwide. For traditional complementary alternative medicine, such as acupuncture, homeopathy, quantum medicine, crystal therapy, chiropractic, pendants, amulets, red thread, molten lead pouring, happy to discuss later, water from mineral springs or holy water, and so on. We had 22 of these practices, and we averaged them. And what we see is, once again, that they're quite widespread, a range a bit higher than in non-adherence from 25 to 45. You see, for example, that China is a bit greener here, but still the takeaway message is that they are mainstream and more of a norm and not an exception. So good news for our research and like bad news for public health, so to speak, but still very important and red flag important to share. Now I'm going to explain which unfounded beliefs we had in mind when we looked into this psychological footprint. So what do people who are prone to these behaviors think and how they see the world? The beliefs we labeled unfounded or epistemically suspect or irrational because they go against principles of normative rationality and ontological assumptions about the world, very abstract feature that they share. And content-wise, they're very different. I will illustrate that. So they're grouped into three categories: paranormal, pseudoscientific, and conspiratorial. Paranormal, we choose extrasensory beliefs as a representative. I believe it's possible for people to sense things from domains beyond their physical senses, or some people can sense future events, for example. And what we see is that people on average agree 3, so midpoint of the scale from 1 to 5. So these are not dismissed as unplausible. They're not given a 1 on average. So these are like quite normalized beliefs. So I'll leave you with that, come back to it a bit later. For pseudoscientific, you have magical health beliefs. For example, incorrect diet makes the food rot in the body, colors change the organism's energy vibrations, plants are living beings whose energy potential can be transmitted to humans, and so on. When we average that, the average agreement with these types of statements is also around 3. So people tend to give it 3, so they do not dismiss it, they do not fully endorse it, but they kind of tell us that they can be possible, okay? And finally, medical conspiracies, the representative of this conspiratorial thing. So for example, cure for cancer has long been found, but the pharmaceutical industry hides it from the public. Or a number of epidemics that are claimed to be caused by natural-born viruses, in fact, are caused by lab-produced ones. Or radiation from mobile phone is detrimental, but also hidden by a powerful telco industry and so on. And once again, around 3, no striking cross-cultural differences. People tend to kind of neither disagree or agree with these statements. What we also see is that these beliefs bundle up together. So confirmatory factor analysis showed us that there is one latent factor and that we have now evidence to claim that there is a worldview it consists of these beliefs. So what does this worldview say? Conspiratorial part kind of challenges the officials, the official epistemic authorities, and it casts doubt at the official recommendations. And then paranormal and pseudoscientific offer alternative, okay? And they kind of function together. Pseudoscientific is especially interesting because it mimics science in its form. So at the same time uses science as a proxy for truth and also diminishes its authority. So I'm happy to discuss these paradoxes. For now, I'm just leaving you with the fact that they do form a consistent worldview. And when we look into relation between those behaviors and unfounded beliefs, what we see is that almost all of our pre-register hypotheses are observed in all the data, both in overall sample and in specific country samples, on average in 32 out of 33. And I cannot tell you how rare this is in psychological research, even though this is correlational, but still we had a couple of challenges, right? So we needed for the instruments to be working and then for the results to replicate in different cultural contexts, but it did happen. So it's like a gem of a data set. I'm very happy with it. And so you see that ESP beliefs, magical health beliefs, and medical conspiracy beliefs are highly correlated with TCAM, and some predictors that you might have bet on at the beginning, for example, education or overall health status, are down there at the bottom of the list. And when we look at intentional non-adherence, we once again see that these unfounded beliefs are important. But also we see that negative experiences with the system, which we also pre-registered because we had that in the Serbian data as well. So that means the experience of discrimination, not being listened to, being dismissed by health workers and so on. So interpersonal experiences, those are also important for losing trust in the system and for not listening to the official recommendations. And down there you see education, socioeconomic status, age, very, very thin or not significant at all. We made the data fully available and we built an interactive application. So if you're like a data freak and like to play with data, we invite you to do that. You can see the frequencies and you can see the relations both on overall sample and cross country. So we would be more than happy if you guys dived into our data. So to wrap it up, what we know so far is that both TCAM and INAR, so both categories of questionable health behaviors, are widespread worldwide. The people prone to one are simultaneously prone to other. And they're very appealing because they're rooted in this set of unfounded beliefs that in turn rely on specific biases. And we also have media to perpetuate these health-related myths and undermine trust in the official medicine. So this is a grim picture with where we are now, but we think that the data that we have can help public communication when it comes to health. So how we as psychologists can contribute? First of all, majority of scientists still subscribe to the so-called deficit model when it comes to communication. So they think it's enough to provide the correct data. People do not know enough. It's enough to provide the correct data, and they will change accordingly. But there is mounting psychological evidence that that is not the case. We also see that scientists think that they should prioritize communication designed to defend science from misinformation and educate the public about science, and not prioritize communication that seeks to build trust and establish resonance with the public. So this top-down elitist approach to communicating science is something that's very dominant in the science community, and we hope to contribute to, to engage in a discussion about this. Where to now? So we thought that our results can be useful for health providers or public health communicators because, first of all, they can think about improving the patient experience because we see that those negative interpersonal experiences are very important to drivers of these questionable health behaviors. And these are, so to speak, easy fixes. So it's not about huge resources. It's not about the, expensive instruments or quality of care. It's about the interpersonal experience. So there is that. Second, you can screen for individuals who are vulnerable to unfounded beliefs that lead to non-evidence-based health decisions, and just see what your patients are actually thinking and how we can maybe discuss or engage with them regarding these beliefs. And we can also build trust in official medicine and challenge unwarranted trust in TCAM industry and media through alternative communication strategies that would go beyond these simple just trust us messaging, beyond this top-down elitist approach to communicating science. As I said throughout, we had all the studies pre-registered and we shared all the materials, data, syntax, and all published articles are open access. So we are like open book and you can see whatever we've done so far, and we're happy to share whatever. Thank you so much for bearing with me, and here is my context and also context of the project and its social media, so feel free to reach out for whatever. I'm happy to discuss with you later.
Wow, thank you so much, Iris. That was amazing. So now we have a few minutes for questions. If anybody has some questions for any of the speakers, this is the time, and you can just pop in questions in the chat or just unmute yourself and ask a question. I might start with one, Iris, and probably because it's the freshest one. Was there one that was was there a result that was the most surprising for you? I did see that gender, I would have thought that education would have had, because again, as you were saying, more of this top-down um, way of thinking, that you would think education would have a more of an effect. And I saw that that one was very surprising for me, but was there one that was very surprising for you?
I mean, it would have been have if we did not have the Serbian data before, where we saw that education was not important at all repeatedly. So it was something that we pre-registered, basically, that like the whole block of sociodemographics would not be predictive, or at least way less predictive than this set of beliefs. So it seems that our educational systems are failing us repeatedly in terms of, giving us equipment to deal with these predatory environment in terms of, media and like biases that they consistently, rely on. So people do not have those, it seems, do not have like strong standards for truth, strong proxies for truth. So they built this worldview that's like, we call it high entropy worldview, where everything is possible, right? So they do not say it like, I subscribe to all conspiratorial claims or all like magical health beliefs in full, but they might be true, right? And just saying that those might be true then undermines whatever you have to, communicate in terms of the official medicine. So it seems to come back to your question that like the education, the whole educational system, even though like critical thinking is everywhere, it's proclaimed to be like an important goal of the system. It seems that it really does not equip us with enough to distinguish between like what's evidence-based and what's not, and so on, and to dismiss some beliefs as fully implausible and just say like, I'm not going to incorporate that in my worldview, so to speak. So yeah, I mean, it's not encouraging, but it's a fact. So we need to deal with that. As soon as we face the truth, I think it's easier to address the problem, I would say.
Yeah. So here's another question, which I am guessing is also for you, Edie, but I guess anybody else could answer it. I think it might be a little bit easier for Liana and Amelia because it was just one language we needed to translate to. But and the question is, how did you translate everything? Oh, it does say Edie's at the end.
Yeah, so we took the procedure from Psychological Science Accelerator. Basically, I was part of a couple of big science projects as a collaborator beforehand, and I saw that that worked. So it was parallel translation by two native speaker, and then a check by a third one to see how that works. So we had basically three native speakers translating and checking the cultural appropriateness of all the questions. Some of the scales were already translated in a couple of languages, but it was like a huge endeavor to translate everything. And we were a bit, hesitant to see how those scales would behave in different cultural contexts, because we did not know 33 languages to basically check ourselves. So we had to trust the collaborators, but it seemed, based on the patterns that we observed in the data, that those worked perfectly. So the translation, it helped that the instruments were not so large. So the whole survey lasted for like 15 to 20 minutes on average. So it was relatively easy for participants and for collaborators as well. So, oops, sorry. I knocked down a bottle, but it's fine.
Any other questions? So if there are no questions, I wanted to share one last thing with all of you, and it is what now? The objective of having these three wonderful guests here is that usually people from the majority world get invited to talk about how horrible it is. to be in the majority world and all of the barriers we have. And I wanted to showcase these wonderful projects that are being done or have been finished and have gotten wonderful results. And so what now is in order to have better generalizability in science is we need to have structural changes. We need to encourage and support big team science led by researchers from the majority world. This is an example from a researcher in Colombia, but this is just one of them. And it was the same way that I joined EDIS and the same way that I joined LAGC, like Diana, and it's the same way that I have joined other projects. So just how we join in big team science, like Many Babies and Psychological Science Accelerator, also support researchers from the majority world. As we know, greater inclusion benefits not only individual researchers, but science as a whole. So this is the only way that we will be able to improve science as a whole by supporting each other and uplifting each other. So if you get the chance, do participate in these studies and support people that may have an idea. For example, the three of you that have been part of these, maybe support somebody else they can do it next time. And with that said, I would like to thank the three speakers for being here. And I will stop recording. And if there's any other questions after I stop recording, we can still do that. But thank you all again for being here. Three very wonderful talks, all very interesting. And I'm very, very excited to hear the results at the end of both Amelia and Diana's project.
Thank you so much. And thank you all the panelists. I mean,
Thank you.
symposium, how do you say that? Symposium participants?
Yeah.
I tend
Oh,
to-
I would like to say super quickly before I stop recording. Same time tomorrow, we will have both Amelia and Edie's and somebody from LAGC and somebody from Many Labs Africa, and we will be talking in a panel about the experience more than the data. So please come tomorrow and have a lively conversation about this as well. And sorry, Edie's, I interrupted you. Go ahead.
No. I just wanted to say exactly that so that we're going to have the opportunity to air our grievances tomorrow. So
Yes.
if you want to hear those, please come.
Perfect. Okay.